Yeah, thanks, Nikolai, for giving me the chance at Evosep, of course, for giving me the chance to talk about some of the research we've been doing in the Target Discovery Institute in Oxford. And I want to start my talk with spinning quite a large circle about talking about location. So I have a couple of slides about location, and obviously every one of us knows that location is quite important. So if you're a policeman or if you're a donut salesman, obviously location is relevant, but also it's of course important of where people are born, where they go to school and so on, and this has massive impact on people's lives. Of course, location can also be quite dynamic. So in this case, for example, it matters where you are in this picture, but location changes, and it's important to also look at trajectories and the perspective. So in this picture, for example, you can think about, okay, where do we start, right, and where do we go? And then, of course, location is a matter of context as well, right? So in this picture, obviously, we can see that you can see or look at this picture with a certain with a certain mindset and say, okay, we see a bus that is drinking water, but that's likely not going to be what is really happening here. So context matters as well. And in the proteomics area or field, we have ignoring these points, so at least when it comes to tissue proteomics for a long time. So when people were running clinical samples and running proteomics analysis, very often they would take punch biopsies, which then are sort of solubilized and analyzed with proteomics, which is all nice and well, but we kind of destroy or ignore the spatial context. And if you think about this, this is also what's happening even sort of in more simplified experiments, right, where we don't have a complex biological system in the background, for example, in a tissue culture dish, like you can see here, right? We treat this tissue culture dish basically as a one-dimensional entity, right? So we just have cells. We completely ignore if a cell is sitting in the center of the dish or at the sort of rim here. And I just want to show that this actually does matter. So when we take or make a very simple experiment here, where we do mass spec analysis of cells that are growing in these different areas, A, B, C, D, E, we can these days very easily identify 8,000–9,000 proteins from these cells. And if we then just look at proteins that are consistently identified in all of these 5 out of 5 samples, then we can look if there are actually differences between, for example, cells that grow in area A as compared to cells that grow in area E. And we've done this in a very simple experiment, and you can see if we just make a scatter plot plotting A versus E, you can see that you get a distribution of proteins that you would expect actually in a much more complex experiment, not from cells that are isolated from the same dish, essentially clones, and which is in this case also a cell line. So you would expect maybe a more consistent proteome across this area. Again, if we do like a very one-dimensional principal component analysis, we can see that sample A and B seem to be very similar, while E and D are also quite similar, and C is somewhere in between. And the differences here explaining 75% of variability. Principal component 2 only explains about 8% of variability. So the location seems to be the driving factor of the differences between the proteomes in these cells that we can see. When we look at the proteome from the perspective of sample A, we can already see that we have quite a nice or wide distribution of trajectories in these proteins. And we can, of course, cluster them and see, look for clusters that kind of behave in a similar way. For example, things that are more abundant in area A and B and less abundant in D and E, and also the other way around, of course. When we then look, okay, what kind of pathways are actually involved in these differential present or expressed proteins in these areas, we can find that, for example, in A and B, we have more proteins involved in OXPHOS, also more proteins involved in extracellular vesicles, but also cell-bound vesicles. GTP binding functions are upregulated here. And for example, we see serine and threonine kinase activity as well upregulated than in the D and E central areas of the tissue culture dish. This all is really just to explain that location matters, right? And that if we see these kinds of effects in a very sort of one-dimensional sample like a tissue culture dish, this is going to be much more complex when we go into the tissue itself. So, and how we can dissect this is essentially we have to segregate areas of interest, and we can do this very nicely and easily by laser capture microdissection. I hope this video works. Yeah, this just shows you basically how a laser can be used to cut out an area of a tissue block or tissue slice, to be more exact. Usually we use a 10 micron thickness here, and then we can isolate the material from this laser dissection and then analyze it by proteomics. We've done this already in 2015, so a couple of years ago, and at the time we had an Orbitrap Velos, and we would compare, for example, cells isolated or cut out from a 10 micron slice in the CA1 and CA3 region of human frozen hippocampus. So here we would, or we cut essentially 85 cells out from these two different areas. Again, these slices are only 10 micron thick. These neurons are quite large, so they have a diameter of 100 to 200 micrometers. So if we think about what we are actually cutting out here, when I say 85 cuts, these are only 85 sections of cells. So overall volume possibly in the range of about 10 cells in total. And these samples were then analyzed by an Orbitrap Velos at the time, and we were quite happy to find 500 proteins that we could quantify across these two different areas. So that was a very nice first initial proof of concept for us, that we could also distinguish the two areas, even with a shallow protein like 500. So that was a good start. Couple of years down the line, we optimized our protocols and could, of course, acquire more sensitive equipment at the time. This is already also like 4 years ago. But what we did here was then cut out cells from certain cell types in human brain. So you would cut out Purkinje cells and Bett cells. These are both cells that are involved in motor coordination. And you can see here basically the area that we used. Purkinje cells are quite common. You can see them here with the arrows, while Bett cells are very rare. And these cells, pre and post LCM, were then isolated, lysed, and then we used an SP3-based methodology that we adopted to our needs and analyzed the samples. Again, this was, I think, on an Orbitrap Fusion Lumos at the time, and that would allow us then to get a meaningful depth of about 4,000 to 5,000 proteins in these different cell types. And again, we had about 100 cells, 80 to 100 cells from both cell types. And we could find definitely certain profiles that are different between these two different cell types. The Bett cells, very rare, were not described for a proteome at that point, while Purkinje cells are quite common and have been described for the proteome and other omics technologies before. So here we could again decide or see that we have proteome, deproteome changes that would allow us then at that point, not only with a shallow depth of 500, but with a greater depth of 4,000 or 5,000, that would allow us to actually draw conclusions about the pathway activities that are distinct between the two different cell types. I don't want to go into much detail, however, I just want to highlight that this approach is kind of a targeted approach where we have features that drive the isolation, in this case, different cell types. And spatial proteomics has been used in a feature-driven form by multiple groups in the last couple of years. And this would allow us then, for example, to distinguish the proteome of an area of a plaque or a tumor as compared to its surroundings, or even do this on across different patients. However, if we keep in mind what I just initially said with the tissue culture dish, it matters if, for example, a Purkinje cells are sitting in this little square, and if it's sitting another one in this little square. So we would expect them to be different. So ultimately, this makes a case for single cell proteomics, but also for a systematic approach for spatial proteomics. And this is what we've taken. And I will explain you why the single cell approach is not really yet feasible in a couple of minutes. So here we would apply a grid to the tissue that we are looking at, and then would do a systematic analysis of the deep proteome. So this is good in a way because we don't introduce bias here, right? So we treat every voxel that we cut out the same way, and we are not saying, okay, we have a feature that we use, a tumor area, for example, that we want to compare, which would limit us in a way that we would only ever be able to distinguish differences between that tumor area and whatever we use for comparison. With a systematic approach, we can discover new areas that maybe a pathologist would not be able to distinguish, or even by immunohistochemistry, you would not be able to say this is a different area from the neighboring area. Ultimately, the goal here is to generate maps. So if we have, let's say, 5,000 proteins in the data set, that means we can generate 5,000 maps across the tissue space that we are looking at. And we used as an example here this ATRT tumor, which is luckily a very rare but aggressive tumor in children. It has basically a SMARCB1 inactivation. So that means apart from this, there's almost no mutational burden. That means usually the immune system in these tissues is not really active against the tumor. So this makes it really hard to treat and usually results in a fatal outcome. So here you see basically an H&E stained slice of this fresh frozen tumor. You can possibly see here now this kind of more solid area in the middle. We have a little bit of clotted blood here, and also this periphery, which will contain cancer cells, but also normal cells of the brain. What we did, and this was just published 3 weeks ago in Nature Communications, we annotated the area using our grid approach, cut out individual voxels from 10 micron thick slices, and analyzed each of these voxels individually by mass spec after SP3 driven digest. So in each of these voxels, then we identified about 5,000 or so proteins, and then we can use the intensities of these proteins to generate maps. And you see here 3 of those. And these maps then basically have a certain resolving power, right? So we did this approach on a sort of the whole section using a limited resolution, a coarse resolution of 833 micron by 833 micron by 10 micron. This was the voxel size. And here we would have about 6,000 cells and would get to a depth of 5,000 proteins in the tissue. Then we also selected an area of interest that would contain the solid tumor core and the more peripheral tissue. And there we went in with a higher resolution of 350 micron as the voxel size. And here we would have about 1,000 cells in this area, and we still would be able to quantify about 4,000 proteins. And then we used one of these voxels and subdivided this into a 9x9 grid. So we have 81 samples here, and this would then have a resolution of 40 micron, again 10 micron thickness, and that would represent about 10 cells, and we would go to a depth of about 1,500 proteins at that kind of resolution. And this is how this looks like. So we have the fresh frozen tissue here, have the grid annotated, and then would just use the laser capture microscope to individually cut these things automatically, followed by sample preparation. So in this case, we made it as such that we would end up with a 384 plate of samples that we then would analyze in mass spec. So we have about 5,000 maps after this. This is the coarse resolution, and we have now here just 4 of those shown. And you can see, for example, hemoglobin here nicely correlates with where the blood clot is in this tissue slice. We can also see that the histone as a proxy for cell number is relatively uniform, with the, of course, exception of the blood area. But we can also see the presence of certain potential markers for the cancer area in the center of the slice, so glycogen phosphorylase. Interestingly, we find the liver isoform, which you would really not expect to see in healthy brain. But we also see sort of a decrease in the abundance of peripherin, which is a neuronal cell marker that seems to be more or less absent in the center where the solid tumor is and still present in the periphery. What we can then do with this data is we can do a clustering. So we can just really do look at basically the profile in each of these pixels or voxels and compare it to all the other voxels in the data set. So this is a spatially unaware clustering, and that would potentially allow us to find new areas in the tumor that we were before not able to see at all. And this is the case when you look at, for example, area 1 and 2 here. And you can see that these are quite distinct, right? And even with the spatially unaware clustering, we see that this we have a set of proteins that distinguish area 1 and 2. And that tells us that there are differences in this tissue. If you would ask a pathologist, and we did ask them, and they told us, oh, you don't expect many differences here because this type of tumor is very uniform, it's very homogeneous. But we have proven him wrong here in this case, that we could find protein profiles that distinguish these areas. And as I said, this is kind of the highlight of the unbiased approach that we are using instead of a feature-driven approach, which would not be able to tell us these differences unless we have a suspicion that there is something like a border between these two areas. Interestingly, we found also quite a lot of immune cell markers, very localized, very focused in this area 13 here and nowhere else. I mentioned before that this type of tumor is a cold tumor, so the immune system is not very active against it. Well, it turns out it is active, however, only in a certain area. That, of course, is an interesting finding that we hope to follow up later on. So this coarse resolution did not give us enough detail, for example, to pin down to localized blood vessels, right? This for me was one important point, because we wanted ultimately to find proteins that sort of correlate with the presence of blood vessels and with that correlate with the abundance of oxygen or nutrients. So we honed in on a certain area, this one here, which kind of covers the solid tumor core, but also the periphery here. You can see sort of the border in the magnified version of the screen, and you can also appreciate that we have blood vessels in here that are quite easily detectable by eye. So this is the medium resolution, and this area was then subdivided into 96 voxels, comprising about 1,000 cells per voxel. And again, we got like 4,000 to 5,000 protein maps here. Here we see again the one of hemoglobin, and you can see that the abundance of hemoglobin kind of correlates with where we see blood vessels in this area. So that means we have now high enough resolution to detect the blood vessels. If we look at the same markers as before, we can see that histone here is really quite uniformly distributed, again, indicating that we have roughly the same cell numbers in these areas or in these individual voxels. And if we look at peripherin as a potential marker for sort of a normal tissue, we can see that's basically absent in the solid tumor area, while it's kind of still present in the periphery. And the potential glycogen phosphorylase cancer marker, which also, by the way, is in discussion to be a marker in colorectal cancer, is quite highly abundant here in the center, or sort of in the solid tumor core, and completely absent in the periphery. You may appreciate that we also actually can see now here potential cancer cell invasion with the normal marker peripherin retreating from this border between the two tissue types, and the cancer marker invading this area. And this just means that we have cancer cells coming into the sort of more periphery of the tumor. Then we went ahead and looked at an even higher resolution. So here we looked at a 40 micron voxel size, and this would only comprise about 10 cells. We picked this area because it shows nicely the presence of four blood vessels, so you can see them here. And we can also identify single out individual cells. So what we did here now was that we sort of annotated each cell and color coded them by their distance to the next blood vessel. So the brighter the color is, the more close, or the closer these cells are to the blood vessel. And with that, the closer they are to the source of nutrients and oxygen. So doing that, we then, of course, did not get such a depth in the proteome as with the larger voxels. So 1500 was sort of the average depth that we received. But we can, of course, now look at proteins and if they correlate with the distance to the blood vessel. And we would expect exactly that to happen for hemoglobin, as it is going to be within the blood vessels. And you can see that's more or less absent in the darker areas, which are more distant from the blood vessels, and obviously highly abundant within the blood vessels themselves. And this is the case also for example, alpha-2M, which is also detectable easily in blood. So now, of course, we are after proteins that do exactly the opposite, right? So proteins that kind of anti-correlate with the distance to the blood vessel. And here's one of those, so ASP-H. So ASP-H has been discussed to be an hypoxia marker. And you can see that this is highly abundant where the darker areas are when we look at the proximity, and lower abundant where the blood vessels are. So potentially now we have a protein marker that can track the abundance of oxygen within the tissue. And you can see that these differences are on a quite fine, small level, right? So each voxel size here is 40 micron. You can now simply estimate what the distance between high abundance and low abundance of this hypoxia marker is. And with that, we have a proxy for oxygen gradients in the tissue. So just a couple more slides, and this is kind of more like a discussion point maybe for later on. So there are issues with this type of precision, because in the review process, we were always asked for this paper. We were always asked, okay, this is great, but the resolution is not fantastic. So in spatial transcriptomics, we have potentially single cell resolution. So what's your answer to that? Well, our answer to that in the paper was adding this kind of information to the manuscript, which I think was a key part to getting this paper published. However, if we do the math here a little bit, we can easily say, okay, in the sort of coarse resolution, we already have 400 mass spec samples to run. With the medium, it's 100, and with the even finer resolution, it's 81. So this is still relatively feasible with today's proteomics workflows. Was not so much feasible just a couple of years ago, but today it is possible to do this. Now, if we wanted to run the whole tissue slice with sort of the medium resolution, we would end up with 2,166 samples to run on the mass spec. Still doable, more of a challenge, but can be done. And in my lab, we have run batches of up to 4,000 samples in one go. So it is possible, it's a thing, but obviously requires quite a lot of dedication and resources. Now, if you wanted to use the highest resolution on a tissue slice of this size, now you then appreciate basically how possible this might be or might not be. So now we are looking at almost 180,000 mass spec runs if we wanted to do this. And this would take on one system 12 years if we run with 40 samples per day. So this is completely impossible. And even if we would run with 400 samples a day, so 10 times throughput, it would still require one machine to run more than a year. And don't forget, we are only at that point look at a single slice, a single 10 micron slice within the tissue, three-dimensional space. So this is a challenge. And if you wanted to do this even on a single cell level, obviously we have 10 cells and each voxel here, all these numbers have to be multiplied by 10. And then even with 400 samples per day, we would kind of have years and years of mass spec acquisition for a single slice of a single piece of tissue, in a single tumor, for example. And with that, I'm at an end. I would like to thank specifically Simon and Ishin here, who provided most of the data that I showed. You can see here my mass spec lab, which is only 42 square meters large and houses 7 mass spec instruments in addition to a liquid handler, which you probably cannot see in the corner here, and other smaller pieces of equipment like a cell-in-one system here, and we have a fume hood here as well. So it's quite a high density mass spec lab. And yeah, with that, I would just like to thank everyone for listening, and I hope everybody has a quiet and peaceful Christmas time. Thank you.